Multiplication by division. Cell cycle. A series of events from the time a cell forms until its cytoplasm divides. Eukaryotic cells undergo mitosis and/or meiosis.
Mitosis.
There are three phases of interphase. G1, where the cell grows. S phase, where DNA begins to replicate. G2, where the cell prepares for mitosis. The cell spends a majority of its life in the cycle of interphase.
Prophase. This is where chromosomes further condense. One of two centrosomes moves to the opposite ends of the cell. Microtubules assemble and lengthen, forming a spindle.
The next stage is metaphase. Nuclear envelope breaks. Next, all chromosomes are aligned in the middle between the spindle poles. The next stage is anaphase. Sister chromatids pulled apart toward opposite spindle poles.
Next is telophase. This is where chromosomes arrive at opposite spindle poles, nuclear membrane reforms, and the spindle fibers break down.
During cytokinesis in an animal cell, the cell pinches into two separate daughter cells. In a plant cell, a cell wall forms in the middle of the cell, and then that splits it into two daughter cells.
After mitosis is meiosis. The first stage of meiosis is prophase 1. That's when the chromosomes condense and the nuclear envelope breaks down. Crossing over occurs. Crossing over is the exchange of genes between homologous chromosomes, allows variation of traits by mixing chromatids.
Next is metaphase 1. This is where homologous chromosome pairs are aligned midway between spindle poles.
In meiosis 1, anaphase 1, the homologous pairs separate and begin heading toward the spindle poles. Homologous chromosome pairs are pulled apart rather than the sister chromatids in mitosis.
Next is telophase 1. That's when chromosomes gather at the poles of the cells and the cytoplasm divides.
Next is cytokinesis, where the cytoplasm divides, followed by meiosis 2. Meiosis 2 is similar to mitosis. Prophase 2 is the first step where the new spindles form around chromosomes and envelope breaks down.
Next is metaphase 2. This is where the still duplicated chromosomes are aligned midway between poles of the spindle.
After that is anaphase 2, where all sister chromatids are separated, pulled apart just like mitosis.
The last phase of meiosis 2 is telophase 2. This is where 4 haploid nuclei form. The result of meiosis is 4 new cells, each with half the information from the original cell, and all genetically different from their parent cell. This results in genetic diversity. This causes different characteristics that allow us to adapt to our surroundings, diseases, and other things.